Tasmanian tigers once roamed the small island of Tasmania, just 240 kilometres off the mainland of Australia, separated by the Bass Strait. The Thylacynus cynocephalus was hunted to extinction in the early 1900s by European settlers owing to their fear of the strange dog-like creature. It was light brown in colour and had several black stripes running down its back. However, despite its appearance, the thylacine had a quiet, reclusive nature and avoided interaction with humans, with an average lifespan of only 5 to 7 years in the wild. So what if there was a possibility of reproducing the DNA function of a genome from the extinct Tasmanian tiger? On May the 21st, 2008, Andrew J. Pask, Richard R. Beringer and Marilyn B. Renfrey published the experimental article Resurrection of DNA Function in Vivo from an Extinct Genome. This investigation turned away from the previous studies of analysis of mitochondrial DNA and focused instead on genomic DNA in an attempt to restore to life a fragment of the Tasmanian tiger genome. It was found that non-coding mammalian genomes containing important DNA information, and this is what started out the investigation. The method involved taking 4 samples from 4 different Tasmanian tigers, 3 of which were taken from fetuses preserved in bottles, and the other from dried skin. The enhancer element of the gene COL2A1, officially named collagen type 2 alpha 1, was isolated from Tasmanian tiger samples and micro-injected into the pronuclei of 4 mouse zygotes by the method of transgenesis. The gene, collagen, was chosen for its chondrocyte-specific expression in the mice. The technique of using a live organism, called in vivo, as opposed to in vitro, where research is done in a controlled environment outside of an organism. The results of the investigation by Pask, Beringer and Renfrey turned out to be very conclusive. The collagen genomes obtained from the Tasmanian tiger samples were found to be in the developing mouse chondrocytes, where it helped to develop cartilage. This image shows the mouse fetus, where the developing cartilage is outlined in blue. This concluded that a non-coding DNA fragment from the extinct Tasmanian tiger could be resurrected or expressed in a fetus using in vivo techniques. The experiment completed its objective of reviving the collagen gene from the thylacine samples. This new information about expressing extinct mammalian genomes in another fetus could become the beginning of gaining more insight into the lives and destruction of extinct species. This investigation and experiment was not in order to bring back extinct species, but to assess gene function and retrieve valuable information. Finally, this exploration into genomic DNA analysis can show that not all genetic biodiversity is lost. Thank you for watching.